Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
As many as half of Americans live with herpes simplex virus, HSV1. Some may be asymptomatic and not know that they are infected, but others develop cold sores and blisters in and around the mouth and can develop blindness, inflammation of the brain, and dementia. How the virus infects the nervous system has remained unclear. In a recent study published in Nature, Greg Smith, a microbiologist and immunologist at Northwestern University Feinberg School of Medicine, and his colleagues discovered that HSV1 kidnaps host motor proteins to move into the nuclei of neurons. The finding has implications for other viruses such as HIV and SARS-CoV-2, according to Smith, and provides an avenue for developing a vaccine. This work is just terrific. It's really important. It's a brand new idea that the virus takes up this motor, and it's needed to get to the nucleus, said Vince Racaniello, a virologist at Columbia University and host of the popular podcast This Week in Virology, who was not involved in the research. But some viruses go into deeper tissues, and in a few cases, they infiltrate the nervous system, typically with devastating effects. But these viruses, such as rabies, can't get into the nervous system on their own. The virus then travels directly into neuromuscular junctions. But herpes viruses are unique. A simple kiss from the grandparents can deliver the virus, which will make its way into the nervous system. If we could understand how it actually does this, we could turn that around to our advantage, Smith said. For example, researchers could use that information to develop gene therapy vectors that target the nervous system and to protect people from herpes viruses in the first place. Smith's team knew that HSV1 enters the nervous system from a nearby epithelial cell and then takes an 8-hour journey from the terminal end of a neuron's axon to the centrosome, a central hub for traffic moving about the cell, carried by motors called dynein. To infect the nervous system, herpes viruses have to deposit their DNA genome into the nucleus through a nuclear pore. But dynein motors only travel toward the centrosome. Smith and his colleagues always assumed that there must be a kinesin motor, which travels away from the centrosome, that helps the virus finish its journey to the neuron's nucleus. To find this kinesin, Smith's team mapped out sites on viral proteins that might interact with the kinesin motor and mutated them. Then they showed that these viral mutants traveled down a nerve to the centrosome, but were unable to make the final jaunt to the nucleus. What we've now learned from the study is that not only was HSV1 making more viruses in the epithelial cells, but those progeny viruses were picking up the epithelial cell's kinesin motor and packaging it into the viral particle itself, so it became part of the virus, Smith said. The virus holds onto the kinesin when transferring into the nerve terminal and during the entire marathon run down the axon, and only uses it when it reaches the centrosome to then travel to the nucleus. What's so cool is that the virus is stealing a locomotive basically, and packing it up in its virus for use in the next cell, said Racaniello. We've never heard of this before. It's unprecedented. Reference C. Pegg et al. Herpesviruses assimilate kinesin to produce motorized viral particles. Nature, 2021. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.